Hi, everybody. And first of all, I want to start by thanking Chris, Giovanni, and Liana for the organization of these great seminars, and also for inviting me here to share the work that we've been doing in the last few years, focusing more. Okay, I'll focus a little bit more on the part that is related with sphingolipids, but also with part of our work that is partially connected with it. So the main question that we've been addressing in my lab is trying to understand how lipids are relevant for brain function, and the strategy that we've been using for that has been using mass spectrometry lipidomics, where we can actually produce a lipidome for a given biological sample, either from a physiological or from a pathological condition. And we've actually addressed this over the past few years, and we've shown that the lipidome of brains from patients with Alzheimer's disease and from mouse models is altered. And we've started to get interested in other factors that affect Alzheimer's disease pathogenesis, such as environmental chronic stress. And what we actually hypothesized was that this chronic stress exposure could also alter the brain lipidome. And for the ones who are not so familiar with chronic stress is any kind of exposure that can lead, for instance, to anxiety-like behaviors, to depression. And in the situations where we model the effects of chronic stress in the lab, using animal models to study it, there are two aspects that are very important to take into account, which is, first of all, the duration of the stress. In this case, for our own purpose, is that it has to be chronic for the effects to actually be noticeable in our models. And the other feature that is fundamental is that it needs to be unpredictable. And this part of it for our models is relevant, especially to observe in the cases of anxiety-like behaviors. And overall, there have been various groups studying this over the past few decades. And concerning the model that we use in our lab, which is the chronic unpredictable stress model, it has been shown that it affects a wide array of phenotypes in rodents. And this also has implications for humans, of course. And for instance, it has been shown to lead to endocrine dysregulation, leading to increased levels of corticosteroids circulating in the blood. And from these, the main one that has been focused as a stress hormone has been cortisol in humans, and in rodents, the equivalent is corticosterone. Another consequence is the reorganization of dendritic trees. Also, chronic stress leads to neuroplasticity alterations, for instance, at the level of long-term potentiation modulation, and also to behavioral alterations, namely at the level of memory and learning deficits, but also in leading to anxiety-like behaviors. And for the first approach that we targeted in my research lab was to actually try to understand the comparison of two different situations: a control situation where the animal is exposed to a control environment, comparing it then to a chronic stress environment, and after the exposure for 4 weeks to these two different behavioral environments, we took kind of a metabolic picture of these situations, and we used lipidomics to actually probe that. And briefly, the model that we use in the lab, this model of chronic unpredictable stress, it is exposed for 4 weeks, and we use one different stressor per day for about 1 hour duration. And this includes shaking, overcrowding, strobe lights, or exposure to cold water or a hot air stream. And these stressors are exposed at different times of the day to actually give that degree of unpredictability to the rodents. And after these 4 weeks, to actually make sure that the animals are indeed stressed, we measure in the blood of the rodents the levels of corticosterone as one of the effectors of this stress response. And indeed, we can see here that the stress animals have increased levels of circulating corticosterone. Then, after this moment, we actually sacrifice the animals, and then we dissected different regions of the brain, namely the hippocampus, the prefrontal cortex, amygdala, and cerebellum, and we performed lipidomic analysis of these regions. Here I'm showing a heat map where in each column we have the different regions, in each line we have the different lipid classes, and whatever it's red, it means that it's going up comparing to control. Whatever it's blue, it's going down comparing to control in the stress conditions. And overall, the first thing that we can see is that the region that's at the lipidome level that is most affected by stress is the prefrontal cortex, and after this it was the hippocampus. Then at the level of the different lipids that are altered, among many changes that we saw, there were sphingolipid alterations, and from these we actually saw a decrease in sphingomyelin and an increase in ceramide. And there is actually an enzymatic reaction that connects these two, which is by a sphingomyelinase. It's one of the possibilities. And this was actually interesting, this possibility, because before our own observations, there was already another publication using other methodologies that showed that chronic stress led to increased ceramide levels in the hippocampus of these mice after exposure to chronic stress. And interestingly, treatment with antidepressants blocked this increase in ceramides. And another set of experiments from this publication that was interesting was that the exposure to corticosterone led to a depressive-like behavior, and mice that were deficient in acid sphingomyelinase were actually resistant to this depressive inducing behavior upon corticosterone treatment. So in our own work, to actually get more mechanistic insights in what was going on, we took into consideration all the animals that we had studied, all the 350 lipids that we had studied in our lipidomic analysis, and the 4 brain regions that we analyzed, the prefrontal cortex, hippocampus, amygdala, and cerebellum. And we plotted these lipids in each brain region to the corticosterone levels in order to understand what would be the lipid species that would be affecting, at least trying to explain how corticosterone would be changing this lipidome in the context of chronic stress. So we would have something like this, a correlation index between the lipid species and corticosterone, and then we can actually plot all these values, these correlation values, and we take into consideration the slope of the curve. We can consider that whatever it's going to the right is more directly correlated, whatever is going to the left is more inversely correlated. And taking a closer look into all this data, we can see, for instance, that in the case of phosphatidic acid, it was a subset of the species were more to the right, which means more directly correlated, and phosphatidylcholine species more to the left. So this means that phospholipase D activity could be actually increased in this setting. Another interesting hit that we found was that a different number of glycerophospholipids were more to the left, and lysophospholipids, at least a subset of them, were more to the right, which indicates that phospholipase A2 activity could be increased in this setting. And finally, we found that sphingomyelin species were more to the left and ceramides were more to the right, which potentially implicates, again, as we had previously, it was already aligned with our previous observations, that sphingomyelinase activity could also be increased in this setting. So in conclusion from this first part, we showed a detailed characterization of the impact of the chronic stress in the brain lipidome. We also provided some mechanistic insights for the effects of corticosterone in different regions of the rat brain. And we identified specific lipid signaling pathways, namely sphingomyelinase, phospholipase A2, and phospholipase D, that could be impaired in a stressful condition. After this, we wanted to take a step back and try to understand what could be the alterations in regions of the brain that could actually be relevant both for Alzheimer's disease and also chronic stress. And taking a look into the literature, the region that actually seems to integrate both the alterations of both these diseases is actually a region called the hippocampus. And the hippocampus, which I'm here representing the rat hippocampus, is located in the temporal lobe of the brain, and it has been reported to be involved in learning and memory mechanisms. And here I'm representing a coronal section of the hippocampus with its different subfields, the data gyrus, CA3, CA1, and CA1 and CA2. And this structural organization is actually maintained along the longitudinal hippocampal axis of the hippocampus. And a number of studies that have actually shown that we can almost segregate the functions of this part of the hippocampus, which we name as the dorsal hippocampus, comparing with the other pole of the hippocampus, the ventral hippocampus. And many groups have been addressing this question. For instance, one of the strategies has been to lesion either one or the other region, which has led to some observations, namely implicating this part of the hippocampus, the dorsal pole, more in spatial memory dimensions in terms of controlling the behaviors of these animals. While the ventral pole of the hippocampus has been implicated more in emotional-like behaviors. On top of this, it has been shown that along the longitudinal hippocampal axis, that there is a gradient of gene expression. And based on this, we actually wondered, could other molecules, such as lipids, could they also be changing along this hippocampal axis? And to actually answer that, we did a very simple approach. We divided the hippocampus in three parts, in the dorsal, intermediate, and the ventral hippocampus, also based on previous studies in its connectivity and also in terms of its gene expression along this gradient. And then we performed a lipidomic analysis. We also compared this lipidomic analysis with other regions of the brain of these rodents. And we performed this analysis, and here I'm showing the data represented as z-scores. And in each column, we have a different lipid class, and each block we have a different brain region. Here I'm starting to show just the three different parts of the hippocampus: the dorsal, the intermediate, and the ventral hippocampus. And just by glancing at it without going into a lot of detail, we can see that the dorsal and the ventral hippocampus are more similar to each other in terms of its lipidomic composition, while the ventral hippocampus is more dissimilar from these two. And actually, when we compare with other regions of the brain, namely the prefrontal cortex and middle and the cerebellum, they are actually more dissimilar from the hippocampus as a whole. Then, to actually focus on our key question that we are trying to address here, we compare directly the dorsal to the ventral hippocampus. And this is what I'm showing here. In black, we have the dorsal hippocampus, and in red, we have the ventral hippocampus lipid levels relative to the dorsal hippocampus. And in each of these columns that you see here, you see different lipid classes. So just looking overall, we can see that there is an overall differential sphingolipid regulation between the dorsal and ventral hippocampus. We could also see that there is a differential glycerophospholipid regulation between dorsal and ventral hippocampus. And then at the pathway level, we could actually explore a lot of these potential differences, but this was actually interesting due to our previous work. We actually saw that in the dorsal hippocampus, there were higher relative values comparing with the ventral in the phosphatidic acid and lower levels of phosphatidylcholine. And there is actually an enzyme that regulates this reaction, which is called Phospholipase D. So from this part of the talk, we saw that each brain region presents a specific lipid composition, that the hippocampus can be subdivided in distinctive structural and functional regions, also based in its lipid composition. And we could actually identify potential pathways that could be differentially regulated along this longitudinal axis. So this actually led us to other questions, and in this case, trying to understand what could be the role of, in this case, this enzyme called phospholipase D in hippocampal function regulation. And PLD is actually one of the main sources of phosphatidic acid. And phosphatidic acid is at the center of a very complex lipid network. In mammals, there are two main PLD isoenzymes, PLD1 and PLD2, that structurally differ by the presence of a loop region in PLD1. In terms of their reaction, in normal conditions, PLD uses water to produce phosphatidic acid. However, PLD has this very, very particular characteristic that in the presence of a primary alcohol, such as ethanol, it preferentially uses it to produce a specific lipid called phosphatidylethanol. And this has been used, for instance, to measure PLD activity in vivo. And some researchers in the past have also used this as a way to indirectly inhibit the function of PLD by diverting its normal production of phosphatidic acid. In terms of phospholipase D's location in the cell, PLD1 has been reported to be more in the Golgi, secretory granules, endosomes, and autophagosomes, while PLD2 has been shown to be more in the vicinity of the plasma membrane. So to study both these enzymes in the brain, the first thing that we did was to use PLD knockout animal models. So we've used PLD1 knockout animals that, as you can see here, this is extracts from forebrain of these animals, and we can see that they only show PLD2. Here we can actually, in this plot, we can identify both isoenzymes by using an antibody that recognizes the C-terminus of both PLD1 and PLD2. And then in PLD2 knockout animals, we only observe PLD1 levels, while in the double knockouts, we actually have no levels at all of either of the isoenzymes. Then, to actually test if this had a functional implication, we use that property of PLD that in the presence of a primary alcohol, it produces this specific product, phosphatidylethanol. And what we did here was do an in vivo PLD activity assay by injecting animals with ethanol. And the animal basically gets drunk. And then after 1 hour, we dissect the brain, in this case, the forebrain. We extract the lipids, and then we measure the levels of phosphatidylethanol. And here, phosphatidylethanol is represented in these black bars. And we can see that the total PLD activity goes down in PLD1 knockout animals. It also goes down in PLD2 knockout animals. And it's basically absent in double knockout PLD animals. So based on this, we can actually take two conclusions from here. The first one is that in mice, PLD1 and PLD2 are likely the two only PLD canonical activity sources. And the second thing is that it actually, if you add up the decrease in PLD1 knockout animals and the decrease in PLD2 knockout animals, you can see that it doesn't go up to 100. So there is some level of compensation going on here. Then we actually focus on that question that we were actually interested, which was trying to understand what would be the impact of the PLD pathway along this longitudinal hippocampal axis. So we measure the lipidome in these animals in both the dorsal and the ventral hippocampus, which we are representing here. And here we are representing the values relative to the litermate controls of each one, PLD1 knockouts and PLD2 knockouts. So the first thing that we observed here in these lipidomic studies was that while in the PLD2 knockout animals, we didn't see any changes in total phosphatidic acid levels. In PLD1, there was a decrease both in the dorsal and ventral hippocampus. On top of that, in PLD2 knockout animals, we didn't see any significant changes at the level of the lipid classes that we were looking into. Then concerning PLD1, we actually saw some other differences at the level of the lipid classes that we were focusing on. And I would highlight these changes in lysophospholipids in the dorsal hippocampus of these animals. We also looked at the species of phosphatidic acid levels that we actually observed, so in a more granular way. And we actually saw that in dorsal and ventral hippocampus in PLD1 knockout animals, they were actually, most of them were actually decreased, while in PLD2 knockout animals, only a subset of these species were decreased, but only also in the ventral hippocampus. And interestingly, no reductions were observed in phosphatidic acid species in PLD2 knockout animals, while we actually saw an increase in one of these species, which further suggests that can be some kind of compensatory mechanisms going on here. So based on these lipidomic results that we got here, we then focus our further studies that I'm going to show here on PLD1 knockout animals, since at least comparatively with PLD2, they were the major source of phosphatidic acid in the hippocampus. And our first approach here was to perform an array of different behaviors that at least and partially rely on the hippocampus. And while I'm not going to go through them all for the sake of time, I'm going to focus more on the ones that we actually saw differences. And just to summarize, we didn't see actually upon PLD1 ablation changes in exploration behavior using the open field paradigm, no changes in anxiety-like behavior in the elevated plasmase, no changes in fear memory, no changes in spatial memory using the Morris-Water maze, and no changes in these more general sociability studies that we've done in PLD1 knockout animals. So the first test where we saw differences was this test called novel object recognition. And basically what we do here is that we place a mouse in this box, and the mouse has time to interact with two objects that are very, very much similar. Then the mouse gets out of the box, and after 1 hour, it's placed again. But now there is a new object in the box. Mice are generally very curious, so they have the tendency and the interest to interact more with a novel object. And this is what we see here in the wild-type animals, where wild-type animals interact more with a novel object. In PLD1 knockout animals, what happens here is that the PLD1 knockout animals spend more time than here with a familiar object. And when we calculate this metric of discrimination between these two objects, we actually see that there is this very subtle deficit that tests the accuracy of this task. Then we also tested this specific social memory task that relies in the hippocampus. And briefly in this task, the mouse is allowed to interact for habituation purposes in these 3 chambers. Then the chambers have like these cups where afterwards we actually are placing the mice there. And the 2 mice that we place here is a mouse that is a litter mate, that the mouse that we are testing knows this mouse for a long time. He has lived with this mouse. And this other one is a new mouse, a stranger mouse that he has never been interacted with. Again, wild-type animals have the tendency to interact the most and the more time with the new animal, because they are curious, they are social animals. Interestingly, in the PLD1 knockout animals, they also interact more time with a novel animal. However, when you take into account all the time that they interact, PLD1 knockout animals are actually less social in this task that actually needs the hippocampus to be properly functioning. So afterwards, we wanted to test the effects of PLD1 ablation in the plasticity paradigms. And here we use two different tests, electrophysiological approaches. One was long-term potentiation, LTP, and then also long-term depression, LTD. And both these metrics are associated with neuroplasticity and have implications for learning and memory. And to do this, we basically perform and prepare these coronal sections of the hippocampus. Then we stimulate in the Schaffer collaterals and we record in CA1. When we give high frequency stimulation, we have LTP. When we give a low frequency stimulation, we have LTD. And we did this both for the dorsal and ventral hippocampal slices. And what we actually observed was that while in long-term potentiation, we didn't see any changes upon ablating PLD1 in either the dorsal or the ventral hippocampus, when we ablate PLD1 in the dorsal hippocampus, we actually see a reduction in LTD induction in the dorsal hippocampus and no changes in the ventral hippocampus. This was actually interesting because during the revision process of our manuscript, there was a paper that came out showing the same phenotype, but in a different brain region in the prefrontal cortex. And another previous paper from the literature had also shown that long-term depression was necessary for novel object recognition. So this actually ties up pretty well both our behavioral and electrophysiological observations. And then finally, we looked into synaptic proteins that could also be associated with some of these phenotypes. We looked into presynaptic and postsynaptic proteins, a different number of them. And from all the ones that we looked into, the changes that we saw upon PLD1 ablation was that there was, in the dorsal hippocampus, a reduction in SNAP25 and a reduction in GLUN2A. And from these two, the one that connects the most with the observations from the literature was SNAP25. And the SNAP25 ortholog that is called SPO20 in yeast is known to interact with phosphatidic acid. We actually use this probe to study phosphatidic acid localization. Also, another paper showed that the treatment with a blocking peptide for SNAP25 reduced the induction of long-term depression. And finally, animals that have reduced levels of SNAP25 show deficits in object recognition and in social associated behaviors. So in summary from this, we saw that PLD1, at least comparing with PLD2, is the major source of phosphatidic acid in the hippocampus, and that PLD1 knockout animals show behavioral deficits in recognition, memory, social exploration, deficits in long-term depression in the dorsal hippocampus, and decreased levels in SNAP25 and GLUN2A levels in the dorsal hippocampus. So as future directions, what some of them we are actually already working on, we are also doing similar studies for PLD2 knockout animals. We would love to go more deeper in some of the hits that we found concerning single lipid metabolism and study its role in the longitudinal hippocampal axis. Also, one of the limitations from our studies would be complemented by doing cell-specific lipidomics for different brain regions. And we are also looking into other neuropsychiatric disorders. The one that we've been focusing the most on currently is also Alzheimer's disease. And just in a nutshell, what I showed here was that chronic stress led to lipidomic alterations in different regions of the brain. If we divide the hippocampus in three parts along its longitudinal axis, we see also specific lipidomic signatures that have implications for behavior. And finally, along the signatures that we found in this longitudinal axis, we found that phospholipase D could be a potential player in regulating these functions. And by ablating PLD1, this had a preferential impact in the dorsal hippocampus. So I just want to thank all the people that collaborated for this work. mainly my former student that I still collaborate with, André Miranda, Luisa Santamarinha, my current student, who's defending her PhD thesis in 2 days. And all the people from ICBS, also collaborators from Colombia. Many of them are not there anymore. And also Marcus from Singapore in the early parts of this project when he was collaborating in the lipidomics analysis.